So it's my honor today to introduce and invite Dr. Ron Breaker as the Life Science Keynote speaker onto the stage. A couple of words about him. Dr. Breaker is the Henry Ford II Professor of Molecular, Cellular, and Developmental Biology at the Yale University, and is an investigator of the Howard Hughes Medical Institute. His lab is working to discover RNA molecules that function like proteins in bacteria and other organisms. Some of these RNA appear to be of ancient origin and perform fundamental tasks such as molecular sensing, gene control, and enzyme functions. Technologies developed by Dr. Breaker's lab have formed the basis of launching two companies, one here in Cambridge, Massachusetts called Archemix, and BioRelics in New Haven, Connecticut. Today, we are looking forward to learning about the latest discoveries and the newest technologies in the emerging world of RNA.
The best hypothesis for how biology began is that it began without the aid of proteins, without the aid of DNA, that chances are it came from a time at which all of the simple biology that was around was being run by RNA molecules. All right? And so that was the premise that I had coming in. That was the hypothesis that we had coming in when I started my lab at Yale now about 17 years ago. And so if you want to study these early, long extinct life forms, you can't study them since they're extinct, and many of their bits and pieces, many of the components that made those simple organisms work are probably also extinct. At least that was our thinking about 17 years ago. All right, so we wanted to look at whether there was an RNA world, in other words, a biological system run entirely by RNA, and if there was, could we sort of reinvent some of these long-lost artifacts of an RNA world? So I show you this. This is a cover of a book called The RNA World. And in this book is a series of chapters that explain to scientists why, what is the evidence that life began or passed through a time which everything was done by RNA. And I put a few things on this list. I won't go through them all. Just want to note that one of the most compelling reasons why we think RNA came before proteins is that the machine in all living systems that makes proteins is an RNA machine. And the machine is called a ribosome. It's a very big machine. I'll show you a picture of about half of the ribosome in one of my slides. This machine is largely made of RNA, and at its core, where amino acids are stitched together to make proteins, to make enzymes and other structural proteins and things that you need to run a modern cell, at that very center, there's an RNA machine putting together those amino acids. And the argument is that the last thing you would expect for biology to choose as the machine to make proteins is an RNA machine, unless RNA came first. Okay, so that's one of the compelling arguments that is used to argue that, hey, at one time RNA was running the entire show. All right, so if that's true, then we would argue that there are a number of widgets, there are a number of devices that RNA world creatures would have needed, and that some of these things are probably extinct. But perhaps some of these things are around still in modern cells, like the ribosome. If the ribosome was this machine making proteins at the very beginning, it's still with us today. Maybe there are other lost RNA world relics, molecular relics, that we can find and understand how life was billions of years ago. Equation 4 to the n allows you to calculate how many different RNA or DNA molecules you can make. Right? So there are 4 different bases in DNA, G, A, T, and C. And if you have only a DNA of only one link, one nucleotide in length, you only have 4 possible DNA molecules you can, that you can make. Right? So it's N is 1. So you have G, A, T, or C as the 4 molecules. All right? If you have a chain length of 3, you have 64 possible combinations, right? 4 times 4 times 4 is 64 possible combinations, right? Biologists love that number because that's the number of different codons in the genetic code. But this number, as you notice, is going up exponentially. If you wanted to make all possible DNA molecules of 85 nucleotides, which is a tiny DNA, 85 nucleotides. The human genome has 3 billion nucleotides. I'm talking here just about a tiny DNA. If you wanted a copy of every possible sequence, you'd have to convert 10,000 Earth masses of material into DNA in order to have one copy each. That's a huge amount of material just to make even all possible combinations of small DNAs. Again, this gets absurd really fast. If you go to 125 nucleotides, this is far smaller than any virus, far smaller than any organism's genome. You would need, well, in fact, there's not enough to sample all possible combinations, even if you converted all the mass in the universe into RNA or DNA. One conclusion is that biology has not fully explored all possible DNA and RNA combinations. Right? No matter what some biologists might tell you, oh, biology has tried everything, and when you look at a modern cell, everything there is as good as it can be. That's completely, utterly false, because there's not enough mass in the universe, or not enough time in the universe, to have sampled all combinations. All right? So that leaves some opportunities for people like me, molecular engineers, to create things that perhaps biology has overlooked or never intended to create. All right, so that gives you sort of interesting technological potential for making things out of RNA and DNA that haven't existed before. The other thing that I want to say is that, again, going back to this idea that at one time there was a biological system made entirely of RNA, that some of those molecular inventions that life has made in the last 4 billion years, that they've gone extinct, and that maybe we can recreate some of these things. We can create these things in a laboratory, much like natural evolution creates them over millions of years. And the way we do this is that you can make a population, a random sequence population of RNA or DNA. Just make these molecules at random. We can make trillions of them in a single test tube in a span of a few hours. Then we have to have a system to select. So the first stage here is to select for those RNAs or DNAs that have some special property you're looking for. I'm going to show you an example of how we make these molecules break apart. Okay, so we can select for molecules that may break themselves. All right, once we select them, we can then reproduce them or amplify them. So we simply have these molecules making baby molecules, making copies of themselves, and then we can subject them to more mutation if we want. We can introduce changes into those nucleic acids, and then we get a new population. But instead of this being random, it's now selected to have certain functions. All right, so here's an example of what we've made with this test tube evolution approach. We've made DNA molecules. Now remember, these are DNAs. I just told you why these things are inert, why they don't do a lot of chemistry. And yet, even with these, even as inert as DNA is expected to be, we can use these powerful test tube evolution approaches to create DNAs that do things that nature perhaps never intended for them to do. So this DNA here, the green part, is a particular sequence that when it binds to the yellow part, will cut the yellow part. Right? Now DNA is incredibly stable. You put it in a test tube, put it on this desk, walk away, you can come back millions of years from now, and more than half of the DNA is going to be perfectly intact. All right, it's going to be perfectly fine. But we have these DNAs that will break themselves in a matter of seconds. When you put them under the right conditions, these DNAs have the structure needed to accelerate their own self-destruction by many, many orders of magnitude, by billions of fold in speed. All right, so now you have to ask yourself, who cares? Okay, what might this be useful for? So one thing we've learned from this is that, hey, you can make DNA do chemistry. Biology maybe doesn't do this, we don't know, but we certainly in the laboratory can make them do chemistry. So what good is this stuff? Well, we've made things like this. It's a silly little molecular toy, but we've made this little DNA circle. It's a single-stranded DNA, forms a circle, and it's perfectly stable, unless you add a small amount of zinc ion, and then the molecule breaks open and makes a linear DNA. Again, I'm not sure I know what the applications might be, but we can do this. Now, in work that we've done a number of years ago, we built a number of DNA molecules that see other metal ions. And one of them that we made was a DNA that will cut only when it sees lead. Well, now you have an application for that, right? It'd be great to have a DNA that would sense contaminating amounts of lead. So you could test water supplies, food supplies, whatever. And so we published this, but another laboratory at the University of Illinois has expanded on this. So they've used our lead active DNA, and they've also developed one that senses uranium. And it's not that biology cares about uranium, but in the test tube you can create DNAs that will self-destruct when they're exposed to uranium dioxide. And so on the bottom of that slide, the little green glowing wells that you see, those are DNAs that will glow when they either cut themselves with lead or uranium, depending upon what you've evolved that DNA to do. So these guys have now made sensors that they can sell for testing, for heavy metal contamination. So we do this test tube evolution, and we get lots of answers, and I show two of them on this slide. The one on the left, the RNA is perfectly stable, and then you add this compound FMN, and it immediately turns on. So when this line slopes down, it immediately turns on. And on the other side, we have an RNA, different sequence in that boxed area. That RNA is cleaving very fast, it's cutting itself very fast until you add FMN, and then it shuts off right away. This is exactly what you would have wanted an RNA world molecular switch to do, okay? And we're building them in the test tube using simple test tube evolution approaches. All right, we've done a lot of these things. I won't go into this in any detail, but what the data on the left hand shows is that you can make precise switches. So the top one here is the one I just described, and if you look at these little spots on the bottom, F indicates that we're mixing the molecules with FMN, and that molecule breaks itself in half, and you see the smaller part running at the bottom. All right. Now, if we cut off that part that binds FMN and swap it out for other parts, other RNA parts that bind other things, we can change the specificity. So now the RNA responds to a different molecule. Or you change that domain out, you put a different one in, it now responds to yet a different molecule. And so we felt as though, forget about the RNA world for a moment. We thought that, well, maybe these would make great biosensors for small molecules that may be useful for the drug discovery industry or for, again, sensing for toxic molecules, et cetera. This Boston area company called Archimix, which initially is pursuing molecular senses, and they made absolutely wonderful sensors for different compounds. My favorite story from the company is they made a sensor for caffeine because the soda industry, I'll digress here a little bit, the soda industry would like to save money on preparing sodas in bottling plants, and so they may buy syrup from Coke, and they want to dilute it and carbonate it and sell it to you. And the fewer times they have to mix the syrup with the soda, they save on electricity and time. But if they don't mix it well enough, you're going to get a big bolus of caffeine that is in somebody's can of Coke. And so they want to mix it as few times as possible without killing anybody. All right? So they wanted to have a very cheap and easy sensor for caffeine if they got too much or too little. And so Archimix used test tube evolution. They evolved an RNA that would very precisely respond to caffeine. And in order to test it, they would come into the company, they'd put money in the soda machine, they'd buy a caffeinated or a non-caffeinated soda. They'd have to adjust it's too acidic, so they had to adjust the acid level, and then they would use these RNAs, and it would wonderfully report within about 2% error how much caffeine was in the mix. So that's the kind of science that they began with, and these modern RNA switches we call riboswitches. They're made out of RNA, and they act as genetic sensors and switches. And so where do we find these RNA switches in biology? Well, this shows a cartoon of something that sort of puzzled biologists for a number of years. The problem was that biologists would find genes. You could find a gene in the genome, and that gene is going to make a message. It's going to make a messenger RNA. And you can tell, since we know the genetic code, we could tell exactly where that gene coded for the protein. We knew that region. But there were always regions on the flanks that were, sometimes up to 1,000 nucleotides in length, quite long. And researchers for decades called those regions junk DNA. This was just junk, right? Nothing to see here. Don't waste your time looking there. But in fact, the richest hunting ground for us to find natural RNA switches or riboswitches are in these junk regions upstream of where the protein is coded. The black bar here indicates where the protein is coded. There's wonderful sensor and regulatory systems hidden in plain sight in the regions that we used to call junk. Right? This shows a very nasty pathogen called Clostridium difficile. And this cartoon shows all the various riboswitch classes on the outside and what compounds they bind as well. And then on the inside, it shows a bacterial chromosome. That's where the bacterium is storing all of its genetic information. And the little colored flags on the outside show all the genes that are controlled by these RNAs. And I can tell you right now that the compounds that are being sensed are essential. The genes, many of the genes that are controlled by these RNAs are essential. And that brings up the point, why don't we make compounds that trick those RNA switches? Shut off those important genes. Could we kill bacterial cells? So now we're going from thinking about life 4 billion years ago to thinking about novel forms of antibiotics. Could these be drug targets? All right, so I would propose this idea that riboswitches would be drug targets at conferences. And I would finish my talk, and immediately the first hand would go up, and you'd call on that person, and they would say, well, Breaker, if riboswitches are such good drug targets, why haven't researchers found antibiotics that hit them? Right? We would have them. Industry would have found them. Researchers would have found them. And I would say, you're right. It makes sense that we should have compounds that hit these riboswitches, but it sure makes sense also that these should be druggable targets. So finally, I got sick of that question. And we actually then decided to read some papers. And one of the key papers, a paper from 1943, actually talked about this compound on the lower right-hand side, a compound called pyrethymine, made in 1943, known since 1943 to kill bacteria and fungi, and no one knew how it worked. We now know that that compound binds to that riboswitch, shuts off the production or transport of thiamine, another thing that would be in your vitamin pill every day. All right, so indeed, chemists had found compounds. They showed they killed bacteria and fungi, and no one knew how they worked. All right, so in the last several years, my lab got really good at making compounds like this and new compounds. We got very good, I like to say we got very good at curing plastic test tubes of bacterial infection. And of course, no one cares too much about curing a plastic test tube, but everybody cares about curing people or animals. And so we knew that we were not going to have the skill set needed to go from the light bulb to the finished product. And so we put together a company called BioRelics, which is in New Haven. They're fully active. They're staffed with drug discovery professionals. They are going after that nasty organism, Clostridium difficile, and they're in stealth mode. They are telling nobody what they are doing. And the only way you will find out about what they are doing is if one of their consultants breaks their confidentiality agreement and publishes a paper and describes what they're doing. And so you can indeed find this in a 2011 paper, at least a clue of what they're doing. They have compounds, they have novel compounds, and they are indeed killing bacteria in animal models. So it's very exciting. Well, we want to find more things like riboswitches. And we want to find more things that might be relics from an RNA world. And here's the challenge, right? This is part of a ribosome. This is part of that machine that makes proteins. And the red and green squiggles here are the RNA parts. And so that's an important RNA. It's very big and it's very easy to find. But we're looking for tiny things. This is a riboswitch. This little tiny thing is a riboswitch that sees this compound, which I didn't know existed until we found that riboswitch. It's a modified base. It's used by organisms from all three domains of life. It's not so important what it does, but it taught me and it taught others new biology once we found that new RNA. All right, so we want to learn about new and old biology by finding these things. How are we going to find them? The easiest way is to look in the parts of the genome that everybody calls junk, right? So we're ignoring the parts of bacterial genes that code for proteins. We're looking in the junk regions, and we're trying to find novel, highly structured RNA molecules that do things. And so through a very simple sort of search for RNA world widgets, RNA world devices that may be left in modern cells, we learn about a whole world of fluoride biology that researchers were not aware of. We can understand why toothpastes work as well as they do. But then that gives us new ideas for making very simple reformulations of existing drugs that might work as very powerful topical anti-infectives. I should really stop there by saying that as we find riboswitches, we find all sorts of wonderful new biochemical principles, but some of these riboswitches have uses. I briefly mentioned that they're targets for antibiotics. They can be biosensors, but the really, the fun that we have on the basic science side is to learn about ancient biology and to learn a lot more about modern cells and the potential technological benefits once we understand modern cells in a bit better way. I will stop there. I'm just going to show you a picture of the people who really do the work in an academic lab, all sorts of wonderful young people who are not only getting their education, but I think pushing back the frontiers and the mysteries of biology in very rapid ways. I'll thank you very much and be happy to take a question or two.